Hi everyone, so today we're going to be looking at dihybrid crosses, and the key knowledge that you need to know is the predicted genetic outcomes for two genes that are either linked or assort independently. So a dihybrid cross, di means two, so when we use a dihybrid cross we are predicting the genetic outcomes for two traits. And we can predict the genotypes and phenotypes of offspring using a Punnett square, but it's a much larger Punnett square. As you can see on the right hand side over here, we've got two genes. So over here, capital B, lowercase b, and then capital E, lowercase e. Because we're looking at two traits, we can have all these different options. And so I'm going to go through a dihybrid cross. It's a little bit more complex, but we will get there. Okay. So if you were to write out your Punnett square the same way that you did for a monohybrid cross, well, you would be wrong. This is not the way to write it. The correct way to write out your Punnett square is like this, and you can see that there are two alleles for each of the squares, and then you're going to write the combinations you're going to fill it out through here. So this is the correct way to write them. There's a formula that you can use, which I will teach you in just a second. Here we have two cats. Both are heterozygous, as you can see from their genotypes. Capital S is short tail, lowercase s is long tail. So both cats have the allele for both. Capital S, of course, is dominant, so they're going to express the dominant allele as the dominant phenotype. And capital B is brown fur, lowercase b is white fur. Again, both of them have that capital B, so they're going to express that dominant phenotype. In order to figure out how you fill out that dihybrid cross, I want you to use the FOIL method. So you're going to take the first alleles and combine them. So the capital S with capital B. So you follow that red arrow around, and when you would write your first allele combination would be capital S with a lowercase b. Then the O of FOIL is the outside alleles, so capital S, follow the yellow arrow around to the two outside alleles, and capital B. So you write capital S / capital B, and then the I of FOIL is the inside allele, so these two here, so lowercase s and lowercase b, lowercase s, lowercase b, and last the L, so the last alleles, this last allele and this last allele, where you get lowercase s and capital B. So these are the possible combinations that one of the cats could pass on. Because the second parent has the exact same genotype, these combinations are going to be the same. So they're going to have capital S, capital B, oops, sorry, let's try that again, capital S and lowercase b, capital S, capital B, lowercase s, lowercase b, lowercase s, and capital B. So you just follow the FOIL method for the first parent and then follow the FOIL method for the second parent. So these are the possible gametes that this first parent could pass on, and these are the possible gametes that the second parent is going to pass on. And this is what you're going to put at the top and down the side of your dihybrid cross Punnett square, which I will show you on the next slide. As you can see here, I have written one of the parents' possible gametes or combinations of their two traits as alleles down here, and the combination of these alleles up here. Which parent you put on which side doesn't matter, you're going to have the same outcomes in the end. So let's fill this out. All right, we're going to take this capital S and this capital S and put it in here, and this lowercase b and this lowercase b, and put it in the box. So you would write it as capital S, lowercase b, lowercase b. Okay. Then we're going to move to this box over here. So we've got a capital S and capital S again, but this time we have a capital B and a lowercase b. So we go capital S, capital S, capital B, and then lowercase b. Now, you wouldn't be wrong if you wrote the lowercase b first. However, the capital or the dominant allele is usually written first. So that's how I want you to do it. But you wouldn't be wrong if you wrote that lowercase allele first, because that combination would still be the same. So let's keep going. I'm going to keep going down this row. So we've got capital S, lowercase s, lowercase b, and I'm just going to keep going through this and move on to the next row. So capital S, capital B, lowercase b, and I'm going to see me write this out. It's a little bit tedious. Capital S, lowercase s, capital B, lowercase b, and so on. Alright, again to the next row, we got lowercase s, lowercase b, and lowercase b. I'm going to fill it out using these as well. So capital, lower, bear with me, this takes some time. Alright, and now we have our last combination, so lowercase s, capital B, lowercase b, and then so on. So okay, so it looks kind of messy, but it's all in there. So these are all the possible combinations of alleles that these two parents could produce in their offspring for two traits. So dihybrid cross, we're crossing two traits. Now, if you ever are struggling to distinguish between a capital and a lowercase, you can always write the capital as normal, and then you could write the lowercase with a line on the top, or even the lowercase with a line on the bottom to indicate that it is the lowercase letter. If you didn't, as long as I can clearly distinguish between the capital and the lowercase, that's totally fine. I've just typed it all out, so it looks much neater now, and you can see all these combinations. Looking at it just like that seems a bit overwhelming, so I'm going to color code them on the next slide, and you can see the different genotype combinations. And here they are. So you have all of these possible genotypes that could be produced from those parents. And I've put them as a ratio and as a percentage. And I color coded them to do this. So if we're looking at, there is one of the 16 combinations, this one here, will produce a homozygous dominant offspring for both the tail gene and the fur gene. So capital S, capital B, only one out of the 16 offspring could have that, or one of the 16 possible options. So we write it, can write 1 out of 16, and if we did the percentage, it'd be 6.25%. There is 2 out of 16 combinations will have homozygous dominant for the tail, but heterozygous for fur color. And then we can write 2 out of 16, 12.5%, and so on. And you can see we've got all these different combinations. The most common one are these 4 that go right through the middle, which is heterozygous for both traits. And so 4 out of 16, 25%. That means there's 25% chance that the offspring will be heterozygous for both the tail and fur color genes. and so on. And you can look through them and figure it out. Now, this is quite overwhelming to look at when you're looking at genotypes, and a lot of color coding. So when you do this, have a lot of highlighters to color code. Usually though, you're going to be looking at phenotypes rather than genotypes, and I'll show you that on the next slide. Here they are. I've color coded them by phenotype. So all the red squares have at least one dominant allele for the tail. So we'll have the capital S and one dominant allele for fur, and so we'll have a capital B. And therefore, because those are dominant alleles, regardless of what combination of alleles, if there's one dominant S, so one capital S, it'll produce short tail, one capital B, it'll produce brown fur. So 9 of the, that should say 16, not 14, 9 of these 16 offspring will have a short tail and brown fur. Now there are 3 that will have a short tail and white fur because they are homozygous recessive for the fur. And then 3 again out of the 16 will have a long tail and brown fur, as you can see these 3 over here. So they're homozygous recessive for the tail, but have a dominant allele for the fur. And then only 1 out of 16 will be homozygous recessive for the tail and fur. So long tail, white fur. If we were going to write that as a ratio, we'd take the 9, the 3, the 3, and the 1, and write 9 to 3 to 3 to 1, like that. It's a lot of work, and if you take your time and do it properly, you will get this right. What we've been looking at so far is genes that are unlinked, so genes that are on different chromosomes. Now, if we were going to look at linked genes, that makes a dihybrid cross even more complex, because these genes are on the same chromosome if we call them linked, and therefore they're not separated during independent assortment. The gene loci of each is close to each other. So, for example, we have these 2 genes here. It's really unlikely that crossing over is going to occur right in between these 2 genes and separate them. So we call them linked genes because they tend to be inherited together. Not linked genes or non-linked genes are what we've been doing dihybrid crosses on. So 2 genes on 2 chromosomes, 2 traits that can be inherited separately, and what the possible combinations are. Again, if we have 2 genes on the same chromosome but they're really far apart, crossing over is likely to happen in between there, and we call them not linked as well. So to recap, a linked gene is on the same chromosome and is really close together, as you can see here. If they're on separate chromosomes or very far apart on a chromosome, we call them not linked. So crossing over is unlikely between the gene loci of linked genes. We can calculate the chance of crossing over happening using map units. So 1 map unit equals 1% chance of crossing over separating those linked genes. So A and B here are 6 map units apart. They are linked, so there is a 6% chance that gene A and gene B are going to be separated during crossing over. So very small chance. Gene D and E, however, are 52 map units apart, so we're going to call them independent, they're not linked. So there's a 52% chance that crossing over is going to separate D and E. So just to recap what we've learned before, crossing over, if crossing over occurs, as we can see over here, we have recombinant chromosomes. The chromatid that hasn't crossed over, so no crossing over, then non-recombinant chromosomes. So when you're calculating the chance of the genes being separated or what combination of alleles will be possible, we've got to look at map units again. So here we have gene A and B. On one of the chromosomes, they are dominant alleles, on the other chromosome, they're recessive alleles. And they're only 4 map units apart, so there's only a 4% chance that crossing over is going to happen right between gene A and gene B. Because there are 4 map units, we can split that up and say there is a 2% chance of capital A and lowercase b. So that crossing over here and these two parts joining together, there's 2% chance of that. And there is a 2% chance of the alternative happening. So lowercase a and lowercase b joining together and having this combination. It is much more likely, though, that we have just this capital A and capital B being inherited together, or lowercase a and lowercase b being inherited together. So because there is a 4% chance of the recombinant alleles or the linked genes being separated, there is a 96% chance of the genes not being separated. And if we split that into the two possible combinations, we have 48% chance for that happening and 48% chance for this happening. This can seem a little bit confusing, but just look at the map units. So remember that 4 map units equals 4% chance of being separated during crossing over, and then therefore there is a 96% chance of crossing over not occurring between those linked genes. So as usual, bring your questions to class and I will see you there.